So Coccus. And we learned previously that Coccus is a group of bacteria showing the spherical morphology, like balls. And Coccus is a word from Greek, and it means berries. So, okay. The word Coccus comes from a Greek word, Kokkos. It means berry. So Coccus is a group of bacteria similar like round balls or berries. And this group of bacteria contains both gram-positive bacteria, and today we'll cover this part, including Staphylococcus and Streptococcus. So these are the gram-positive bacteria. And we'll also learn a group of bacteria showing gram-negative. That's the gram-negative coccus. So let's see in the genus of Neisseria. And in this group of bacteria, we'll also learn two important pathogens. Okay. So from today on, we'll learn Coccus or other pathogens in a similar pattern. We'll introduce their morphology, culture, and biological characteristics first, and then we'll introduce their pathogenesis, like the important virulence factors or the disease they cause. So then we'll learn how to diagnose infection of different pathogens. So that's a laboratory test people are using in hospitals or in microbiology labs. And we'll also learn the principles of controlling infections. So today we are learning Coccus. We'll learn the principles or methods we can control staphylococcal infections. Okay, Staphylococcus granules. So they are like, you can translate into like grape-like berries, grape-like bacteria. So that's a Staphylococcus. And they are spherical cells, and they are grape-like clusters. And for gram staining, they are positive. They don't have flagella and spores, so which means that they are not motile. So Staphylococcus semen, you want to culture the bacteria, you want to make pure culture of the bacteria, and so that we can further study their characteristics. For Staphylococcus, they are either aerobic or macroaerophilic. Aerobic or macroaerophilic means that they prefer oxygen or they prefer little amount of oxygen. And you can grow them on blood agar. So this is a blood agar medium. In this blood agar, we actually added in blood. So we usually we use fresh sheep blood. So it contains intact red blood cells, and those red blood cells is incorporated into the agar, showing the red color, the blood color. So it contains intact red blood cells, and they have adapted the human body temperature or the animal body temperature, 37. And Staphylococcus aureus, this species can produce pigment, which is a golden color pigment. And except that they are hemolytic. Hemolytic means that they can produce something called hemolysin, and this hemolysin can lyse red blood cells in the agar. We mentioned that the red blood cells are red blood cells. The red blood cells in the agar are intact, And if the bacteria can lyse the red blood cells, it will clear a zone, so the red color is gone, and because of it produce a yellow golden pigment, so this area shows the golden color. So this is the culture characteristics. And for growth characteristics, when you grow the bacteria, when you grow Staphylococcus, it can produce catalase, and catalase is the enzyme which can degrade or split hydrogen peroxide into water and oxygen. And we know that hydrogen peroxide is toxic to bacteria, and the bacteria is armed with catalase, and this catalase can degrade this toxic molecule. And this property, production of catalase, is different from Streptococcus. And that means Streptococcus, which we will learn later today, does not produce catalase. And Staphylococcus can also ferment carbohydrates. They can produce lactic acid, but not gas, which means that when they ferment sugars, carbohydrates, the pH will get lower, so lactic acid will be made. So this is the growth characteristics. And they also express antigenic structures. So some molecules, some proteins expressed by Staphylococcus aureus, like protein A. That's a staphylococcal protein A, short for SPA. It is a surface protein on the bacteria which combines to the IgG1, IgG2, and IgG4 molecules on their Fc region. We learned in immunology class that the antibodies show this Y shape, and this part is the Fc region. And we know that antibodies can mediate phagocytosis. So they can bind, they can recognize the microorganism and bring it to phagocytes like macrophages, neutrophils, and there can promote the internalization of the bacteria into the phagocytes. So that's a phagocytosis. But if the bacteria can produce SPA, which can block the Fc region, the Fc region cannot be recognized by phagocytes. So it will decrease the phagocytosis by binding to the Fc region. And in recent years, another role of SPA is found that they can also bind to the FAB region on B cells. This is B cell, and they have receptors, actually they are antibodies. They can bind to the FAB region, and this will lead to the B cell apoptosis. And when they bind to the FAB region on the B cell surface receptor, they work as superantigen. Superantigen means that they can directly activate the B cell. So that leads to the death. Apoptosis is a type of death of the B cell, and that will result in lower amount of antibody production. So this is a SPA, which can help the bacteria to survive the immune response. And they also produce polysaccharide antigens, and this polysaccharide means like polysugars. They are type specific and also antigenic. The last one is a capsule antigen. We know that some of them can produce capsules, and on the layer of capsule they do have some molecules which are antigenic, so that we call them capsule antigens. which are also specific antigens. This is a form summarizing the characteristics of different species of Staphylococcus. And in this table, the very left one, the Staphylococcus aureus, is the most important one, which are more virulent and cause infection in human and cause us a lot of trouble. They can produce a lot of virulent factors, including like golden pigment, and this pigment can enable the bacteria to survive under oxidative pressure, like the pressure of reactive oxygen species. They produce coagulase. Later we'll introduce coagulase. They can ferment mannitose, they can ferment glucose, and they can produce heat-stable nuclease, and they produce alpha toxin, and etc. So I'd like you to remember the properties of S. aureus, and if you compare S. aureus with other species, you can see that they are structural components. Enzymes. This group of pathogens can produce various types of enzymes, and we'll go through those enzymes one by one. The first one is coagulase. This one we introduced a little bit previously. Coagulase is an enzyme which can convert fibrinogen into fibrin, causing the blood plasma to clot. And we know that in our plasma, in our serum, we have soluble protein called fibrinogen. And these protein this protein is soluble small molecules. Well, it's a big molecule, but small protein monomers. But coagulase can turn this monomer into fibrin, so it's a protein fiber, and causing the plasma to clot. Clot means that it's not like liquid, it's like gel. This will protect the bacteria from phagocytosis, because in the clot, the phagocytes or immune cells cannot swim in. So this make the bacteria safe in the clot. A second enzyme is called hyaluronidase. Hyaluronidase. This is an enzyme which can catalyze or hydrolyze hyaluronic acid. Hyaluronic acids is a group of molecules in connective tissues. And if the bacteria can degrade or hydrolyze this molecule, it can promote the bacteria to spread from one space to another. So this helps the bacteria to spread. You might wonder that the coagulase and hyaluronidase works in a different way and in a conflict way, because if the bacteria produce fibrin, promote the formation of fibrin and lock themselves in a clot, that will inhibit the spreading of the bacteria to other parts. Well, the bacteria are actually clever. They can also produce another enzyme called fibrinolysin. Fibrinolysin is the enzyme which can dissolve fibrin clots. Once they form the clot, and once they feel that it's safe, they will produce another enzyme to dissolve the fibrin and release the bacteria from the clot. So that is useful for spreading. This is the fibrinolysin. And also they produce nuclease. We learned that DNA molecules, and for molecular biology we also we focus on the property of DNA carrying genetic informations in their sequence. But DNA molecules also have have other properties, like they are very sticky, they are very viscous, which means that they can stick other molecules inside the DNA network. So when the DNA is released, they are long molecules. They like a sticky gel, which can stick the bacteria and trap the bacteria. And the bacteria can produce nuclease, which can hydrolyze DNA, so that will break DNA molecules into small pieces. And those small pieces are not viscous anymore, so they are not sticky anymore. That will also help the bacteria to spread from one place to another. So this is nuclease. And also they produce lipase or lipase. Lipase is a type of bacteria which can hydrolyze lipids. And we know that we have lipids on the cell membrane. And if they can hydrolyze lipids, they can break cells, the whole cells, and so that they can interrupt the whole cell's membrane and release the content. And in the content, they might get their nutrients. And also they produce catalase. That's what we mentioned just now. And this catalase can convert hydrogen peroxide to water and oxygen. And hydrogen peroxide is toxic. And once it's converted to water and oxygen, it's not toxic anymore. Okay, so these are the different types of enzymes produced by S. aureus. And also they produce toxins. Toxins are, we are talking about exotoxins. They produce different hemolysins, and these hemolysins can lyse red blood cells. Like they produce alpha lysine, beta, gamma, and delta lysine. And they also produce leukocidin. Leukocidin is a group of toxin which can lyse neutrophils and macrophages. And we know these two type of cells are professional phagocytes, which can eat the bacteria. And they can also produce enterotoxins. And here entero means the digestive tract or the gut. So this is induced when we ingest the bacteria. And in the digestive tract, they produce enterotoxins and induce the central nervous system by showing like nausea and vomiting. And these toxins are heat stable and resistant to the action of gut enzymes. We know that in our digestive tract we have a lot of enzymes, and in the stomach we have a very harsh condition with low pH, and also a lot of enzymes there. But these enzymes cannot degrade the enterotoxins. They are also producing exfoliate active. This is a word sounds like exfoliative toxin. This word means peel off. It's like the falling of leaves. So this toxin can separate the epidermis from the dermis of our skin. It is the principal causative agent of Staphylococcus scalded skin syndrome. This is a very critical syndrome causing serious problem on the skin. It peels off pieces of the skin from the dermis. And later I will show you the picture of this syndrome. Remember that this syndrome is short for 4S. That's Staphylococcus S, scalded S, skin S, syndrome S. So that's short for 4S. And they also produce toxins which can lead to toxic shock syndrome and induce fever, vomiting, shock, and systemic organ problems. Okay. The next one is that they can produce structural components, which are some specific molecules, like they can produce capsules. This will inhibit the phagocytosis by neutrophils and by macrophages, and also facilitate their adherence. So when we introduce capsules, when we learn the general knowledge of bacteria, we learn that they can inhibit phagocytosis and are involved in adherence. And also the structural component peptidoglycan, because they are gram-positive bacteria, they have a thick layer of peptidoglycan, and peptidoglycan have endotoxin-like activity, which can activate the complement system. And also they have teichoic acid, which is also the special component on the cell wall of gram-positive bacteria. They bind to fibronectin and mediate attachment to the host tissue. And also protein A, which we introduced just now. They can bind to the Fc region of IgG molecules and can inhibit phagocytosis. So these are different types of structural components, either on the surface of the bacteria or in the cell wall. Next, we introduce the clinical findings. So that is what type of infection they can cause, what type of disease it can cause. So we call clinical findings. This will include like pathogenic infections and also toxigenic infections. And for toxigenic infections, it is due to the production of different toxins. Now, pathogenic is kind of a symptoms. These pathogenic infections, they can cause different types of pathogenic infections. Some of the infections are just on the surface of our body, like localized cutaneous infections, like infection in our skin. They invade the skin through like wounds, follicles, or glands, so that they can form infection on the surface or on the shallow area of the skin, like this type of infection, or Staphylococcus infection in the hair follicle, or like this one. This is sty. When the kids are young, they sometimes they develop this type of infection. I don't know whether you remember when you were kids, whether you got sty or not, or even when you grow up, whether you got styes or not. That's the sty, the Staphylococcus infection there. And also like skin infection in the skull, in the skin of the skull. This is the back neck. Yeah, so this is a localized cutaneous infection. But the pathogenic infections can also be deeper, so they can infect internal organs like endocarditis. That's endocarditis is the infection in the lung, or meningitis, that's the infection in the brain. Bronchitis, it's the infection in also in the lung. Endocarditis is the infection in the heart. This is in the lung. Pneumonia is also in the lung. So it's deeper infection in other organs, not on the skin. And for the critical condition, they can also cause systemic infection. That means the bacteria spread all over the body. So like they cause septicemia or pyemia. So that's the systemic infection, which is critical and requires intensive care. Okay, second group is toxigenic. That's we call it toxinogenesis. So that's a disease led by toxins. For example, food poisoning, and we know that they can produce enterotoxins, and those enterotoxins will lead to disease, and those disease is characterized by short incubation period. Within hours, they can cause disease, and showing like violent nausea, vomiting, diarrhea, and other symptoms. But there is no fever if it is caused by enterotoxin. And this information will help you to decide whether your patient with like food poisoning symptoms, or whether it's due to infection of Staphylococcus. Another disease is called toxic shock syndrome, because it's a syndrome. It contains different symptoms. This is an inflammatory response syndrome characterized by high fever, vomiting, diarrhea, rash, and hypotension with cardiac and renal failure. So this is a syndrome with a lot of symptoms in different systems and organs. The last one is Staphylococcus scalded skin syndrome, like we mentioned a little bit. That's short for SSSS. That's a 4S. It's due to the toxin called exfoliative toxin, characterized by like peel off the skin, peel off the epidermis. Here is a picture showing this syndrome. It's a threat for the newborn, especially in the newborn. That can cause infection on the skin, and the toxin can cause the peel off of the skin and showing the red area, leaving scalded. They look scalded looking moist area. So this is the staphylococcal scalded skin syndrome. Okay, then we want to learn how to diagnose laboratory in the lab. Well, in the future, you may be a doctor, or you might run a medical laboratory. And if you are a doctor, you need to collect the specimen and send to the lab. And if you work in the lab, you need to figure out what type of bacteria, what type of pathogen in the specimen. And you need to derive the information and make a clear decision what they are. First, we need to collect the specimen. We can use the surface swab pulse, or like aspirate from the abscess, or we can collect the blood, or tracheal aspirate, or spinal fluid. So you collect the specimen from the affected site or the infected site. And once the specimen is collected, we need to send the specimen to the lab as soon as possible. And there in the lab, the people working there will first check their morphology. So that is the first step. We can check by staining the bacteria and examine it with a microscope. So we can use gram-positive. We can use a microscope to see whether they are gram-positive cocci in clusters. If they shows gram-positive clusters, we know that it's some type of Staphylococcus. And you can also culture the bacteria in the lab. We culture the bacteria on blood agar for 18 to 24 hours at 37 degree. When you culture the bacteria, you can also add in different substrates and see what type of sugar, what type of substrate they can ferment. For example, Staphylococcus aureus can ferment mannitose, while other species cannot ferment mannitose. So this will also give you information what type of what species of Staphylococcus is there. And coagulase test. This is the one we introduced before. Coagulase can make fibrinogen into fibrin and make plasma clot. This is positive. If there is coagulase, the plasma will form this gel-like solid structure. It's not flowing anymore. But if it's negative, the plasma is still fluid. It can flow. So we can use this test to check whether the bacteria can produce coagulase or not. Okay, the next is treatment. And after you derived this information from the laboratory test, you culture them, you check their morphology, and check their biochemical properties, and also their enzyme activities. Well, finally you decided that, well, this is a pathogen called Staphylococcus aureus, and we want to treat your patient properly. You want the patient to receive some medicine and to recover as soon as possible. That's the step of treatment. So we need a timely cleanliness and aesthetic management of the lesions. So if you identify the lesion, you need to clean that area, especially on the surface. And if the infection is deep, sometimes we need to open that abscess and release and drain the material in it, the contents in it. The abscesses and other closed lesions, we need to open it with surgical procedure and drain the contents in it. So this is the physical treatment. And then you clean it, you wash the lesion as clean as possible, and then you cannot guarantee that there is no bacteria left there. And there must be some bacteria left. So we need to give your patient proper drugs. So we usually give them antibiotics. But before you give them antibiotics, you need to determine what type of antibiotic is useful, is proper. That means we need to run the antimicrobial susceptibility test. And you want to test which antibiotic will work, and you give your patient the proper one. For systemic infections, that's an infection causing different systems, different organs, and maybe it's pyemia or septicemia, and that's a systemic infection everywhere, spreading. The bacteria spreads to everywhere, and you need to give your patient intensive care, intensive long period of therapy to help them to recover. Okay, so that's what we talk about the Staphylococcus.